Hi everyone, in this video we are going to understand how that sodium dodecyl sulfate can denaturate the protein that we are going to understand from this video. So when we look into the sodium dodecyl sulfate, this is chemical structure of the sodium dodecyl sulfate, which has hydrophobic structure and also the hydrophilic structure. So the sodium part, the group of the sodium part, which has the hydrophilic nature, and the rest of the part, the chain part, has the hydrophobic nature. Okay. Now we are going to see the structure of amino acid, how these sodium dodecyl sulfate can denaturate protein, can be understood by knowing the structure of the amino acid. When we look into the backbone structure of the amino acid, which has two functional groups, one is amino group and another is the carboxyl group. So in between the amino and carboxyl group, we have a carbon atom. So this carbon atom is named as a alpha carbon, because which is more close to the carboxyl functional group. That's why the first carbon atom which is close to the functional group is called the alpha carbon group. We have so many different types of amino acids. In all amino acids, this backbone structure is common, and only the difference in all the amino acids in their side chain structure. So the side chain structure is represented in a red color here. So the side chains, when you look into the side chains, see, these hydrophobic amino acids have side chains. Okay. These side chains have the carbon atoms again, and these carbon atoms are named as beta carbon, gamma carbon with Greek letters. So the carbon atom present in the backbone is named as alpha, and the rest of the carbon atoms which are present in the side chain, they are named as beta, gamma with that Greek letters. So when we see this side chains, so these side chains are different in all these amino acids. Okay. When we look into the hydrophobic amino acids in their side chains, so these side chains have have atoms. These atoms, they share the covalent bond with the other atoms in that molecule. Okay. When we see in the side chains, these atoms, how they share with the covalent bond with the other atoms in the side chains. So we know that covalent bonds are classified into two types, polar and non-polar. In the polar non-polar, the difference is though in both conditions, they share the electrons between the atoms. So covalent bond is seen when the two atoms, they share the electrons, unlike the ionic bond, where the electrons are transferred from one atom to the other atom. So in this covalent bond, so when during the sharing of these electrons, if these electrons are more close to one atom, and they are far from the other atom, so that even inside of this structure, so one atom gets a partial positive charge and one atom gets partial negative charge. So that condition is called the polar condition. So such a bond is called polar covalent bond. When the two atoms, the shared electrons are not close and not far for two atoms, and they're equally distant from the both atoms. So we say that they're covalent non-polar atoms. So we say that's a covalent non-polar bond. Okay. So these side chains, they have such a non-polar covalent bond. That's why, so we say these amino acids are non-polar. So we say they're non-polar amino acids or hydrophobic amino acids. So when such amino acids are present in a protein, so when this protein undergoes when this protein folds to form its 3D structure. So these hydrophobic amino acids, they move inside of the protein. That means the core part of the protein, that means the interior part of the protein, gets this hydrophobic amino acid, and the upper surface of the protein will have the hydrophilic amino acids, because the proteins are present in the cytoplasm, which has water. So the water cannot form bonds with these hydrophobic molecules which are non-polar. So because the water can interact with only polar molecules, so all the polar structures which are present in the other amino acids, so they come on the surface of the protein, and these hydrophobic amino acids are arranged in such a way that they are present inside of the protein. So when we see this alanine or valine or isoleucine, leucine, and this methionine, phenylalanine, tyrosine, and tryptophan all have such type of hydrophobic side chains. Okay, this is about this one. Okay, then now we are going to understand how this sodium dicyl sulfate can how this sodium dodecyl sulfate can denaturate the protein. So the sodium dodecyl sulfate, which has hydrophobic tile, so that hydrophobic tile interacts with the hydrophobic chains of this particular protein. When this hydrophobic chains they interact with this particular hydrophobic tiles of the sodium dodecyl sulfate, so that when it interacts with that sodium dodecyl sulfate, so that these proteins will get a polar structure, because the sodium dodecyl sulfate has, I said, no hydrophobic tail, and also it has hydrophilic head region. So now every protein, that means every amino acid, this hydrophobic amino acid gets a positive charge on it, so that every amino acid becomes a hydrophilic amino acid because of this sodium molecule present in the sodium dodecyl sulfate. Because of this, now these hydrophobic amino acids containing region which is present inside of the protein now can come outside of the protein to have interactions with the water molecules, which have the polar which have the polar nature. Therefore the protein loses its structure, 3D structure becomes, and so loses its 3D structure and becomes non-functional. So I can say it's the structure of the protein gets disturbed when we add the sodium dodecyl sulfate. So this is how this particular sodium dodecyl sulfate can denaturates the protein. The same sodium dodecyl sulfate, because of its charge, positive charge, so now the entire protein gets a charge, and this charge can be used. So this charge can be used to separate even the proteins on the gel. So to separate the proteins on the gel, so the protein molecule must have a charge. So the protein molecule now, because of this sodium dicyl sulfate, it gets a positive charge, so the protein becomes positive. And using this electrophoresis, we can separate the proteins based on their charge now, because all the proteins now have charge, so we can separate based on their size, sorry, not charge. So we can separate them on the based on their size, by using their charge to separate them on the uh, gel, or to separate them on the polyacrylamide gel. Okay, so this is the purpose of using sodium dodecyl sulfate. We can denaturate the protein, and also this same SDS compound may attributes charge to the protein molecule. Okay, the same SDS we use the purification of the plasmid. So why we use for purification of plasmid? So when the plasmids are present inside without desired gene, to extract our desired gene from the plasmid, so you have to isolate the plasmid from the bacterial cell. So since these plasmids are present inside of the bacterial cell, so we need to break down the plasma membrane. The plasma membrane has a lipid bilayer, and also it has proteins on it. So we use SDS as a chemical to destabilize the lipid bilayer, as well as to destabilize the proteins, or I can say that no so proteins becomes, their structure, their structures. So to disturb their structure, we use the sodium dodecyl sulfate, because the SDS has a hydrophobic tail, so that SDS interact with the lipid bilayer, and so that it forms bonds with the hydrophobic tails of the lipid bilayer, so that the lipid bilayer structure gets disturbed. Okay, also the sodium dodecyl sulfate interacts with the proteins, and that confers some hydrophilic state to the hydrophobic amino acids present inside of the proteins, so that the protein denaturates. This is how that proteins becomes denaturated by the SDS, and the hydrophobic interactions inside of the membrane also gets weakened by the SDS. So this is how we use SDS. Okay, this is about the SDS and the protein interactions. So usually we should understand how that covalent non-polar bonds, the covalent non-polar bonds are present in the these amino acids, and how these non-polar amino acids can give a 3D structure to protein by arranging in such a way that these are present inside, or the core part of the protein. Thank you so much.